the labs really quickly before you have some time to look at the lab essays. One thing that I want to remind you that you can look at is a resource called PH Lab Bench. It stands for Prentice Hall Lab Bench. Now these diagrams and essays are based on the old labs that we used to do. I think hearing information about any of the labs is going to be helpful. I do encourage you to go back and read through your lab manual at least once. But using this website can help because it has animations as well. When we look at the lab about diffusion and osmosis, it doesn't matter what data they give you. You want to know that membranes are semi-permeable. And so only selective things are going to be able to fit through. Because most labs that we do are going to focus on passive transport, we're going to be looking at limiting molecules based on their size, but also based on their polarity. So remember, small, non-polar molecules will make it through easily. We do usually have transport proteins, mostly for water. So don't worry about writing everything down. I'll post this if you want. But the key is that you can look at any example of osmosis or diffusion and predict what will happen. So when we give you an example of diffusion or osmosis, you want to know, how do things diffuse? They move from high to low concentration. We can use active transport, but that will require energy. One of the examples we used compared glucose to starch to iodine, and we looked at the end result to see what moved. Now this was the potato lab. We put different pieces of potato in different solutions, and we put different bags in different solutions. So the key that you have to be able to do is you have to be able to predict which way is water going to move. Remember, water moves from high to low concentration. And so you want to try to bring back your words hypertonic, hypotonic. One thing is that we did work in percent change. So we have to keep in mind that if everything doesn't start weighing the same amount, that we do need to use percent change. So when you're looking at this essay, it is asking you how would you set up an experiment. And so you would want to keep in mind that you either want them to all have the same mass, or you would want to use percent change in your calculations. The next lab that goes along with an essay is the enzyme lab. Now we did a slightly different version of this year, and we got mixed results. Keep in mind it doesn't matter what lab or enzyme you're given, enzymes speed up reactions. They do have to fit into the active site, so only if they fit into the active site will a reaction occur. And that means that the enzyme is specific. So you can add different substrates, and only the ones that fit in will actually have a reaction occur. Keep in mind, the more enzymes you have, the faster the reaction will go until you run out of substrates. And so what this lab actually looked at is how quickly will this reaction occur? What is the number of moles? This graph looks at the number of moles of product. Keep in mind, you can measure the rate of the disappearance of substrates or the appearance of products. Normally we'll have a maximum rate at which the enzyme is working, and that's what we call saturated. It means that we can't work any faster unless we add more enzymes. Now eventually the enzymes will break down all the product, and our reaction will slow and stop. Another way to slow and stop a reaction is actually by adding an acid, and so we can disrupt the bonds. So don't worry about the indicator. For the mitosis and meiosis lab, I really encourage you to just go back, read through the lab manual about the cell cycle. You should be able to look at pictures and identify, and you should be able to identify that the cell spends the most amount of time in interphase, and that mitosis is pretty quick, and you should be able to identify the purpose of meiosis. You can always pause the video, but we did also look at the results of crossing over and how crossing over gives us new arrangements of traits. The pigment and photosynthesis lab, this one we did with the discs, and we looked to see which ones would float. Keep in mind that in order for photosynthesis to occur, we need carbon dioxide, and so that was in the solution. we needed water, and we needed light. And we pulled all of the oxygen out, and as oxygen bubbles formed, the leaf discs floated. You can look at all the different pigments, and this is where the video is going to stop.